Welcome to the Molecular Therapy Podcast. I'm Dr. Rory Bricker-Anthony, Scientific Editor of the Molecular Therapy family of journals. This episode features a conversation between Dr. Timothy Cripe, Editor-in-Chief of Molecular Therapy Oncology and Professor-in-Chief of Hematology Oncology, BMT, at Nationwide Children's Hospital, and Dr. Marco Davila, Rustum Family Endowed Chair in Translational Research, Senior Vice President and Associate Director for Translational Research, Department of Medicine and Immunology, Roswell Park Comprehensive Cancer Center. They will discuss a recent article published in the December issue of Molecular Therapy Oncology by Dr. Davila and colleagues titled Bispecific CD33 CD123 Targeted Chimeric Antigen Receptor T Cells for the Treatment of Acute Myeloid Leukemia. As you may know, the ASGCT annual meeting is right around the corner, and the Molecular Therapy family of journals will be on site at the annual meeting for a special Meet the Editors event. Stop by ASGCT Central on Friday, May 10th at noon to meet the editors-in-chief of all 4 journals in the Molecular Therapy family. Our editors look forward to answering your questions and discussing your ideas for journal submissions. As always, ASGCT members save big on annual meeting registration, $385 this year, whether you're attending virtually or in person in Baltimore. Find the full program and register for the meeting today at annualmeeting.asgct.org. And now, Dr. Cripe and Dr. Davila.
All right, Marco, let's jump into it. I appreciate your being here today, and we're here to talk about the paper you published in Molecular Therapy Oncolytics, now rebranded as Molecular Therapy Oncology, regarding bispecific CD33 and CD123 CAR T cells for AML. Why don't we just start by having you give us a brief background about AML and why it's been so challenging?
Sure. So I guess there's several reasons why acute myeloid leukemia has been very challenging for the development of gene-engineered cell therapies. First and foremost, probably should highlight that it's been the most successful therapy for standard allogeneic stem cell transplantations. But when we were developing CAR T cells for hematologic malignancies, CD19 was a great target because we understood the potential for on-target off-tumor toxicities. So that meant that, at worst, we might destroy normal B cells, but felt that that was still, compatible with life. But knocking out myeloid markers, knocking out neutrophils was a major concern. Luckily, there's been great success for B-cell malignancies in myeloma using CAR T cells, but acute myeloid leukemia remains the major hematologic malignancies where we don't really have promising clinical results with CAR T cell therapies. So I decided to kind of take a step back and look at, engineering technologies available to us. How can we potentially mitigate some of the risk associated with acute myeloid leukemia with things such as CAR design? So really the big challenges with targeting AML is target heterogeneity, meaning that there are no targets are present on 100% of AML blasts, and also co-expression of targets on hematopoietic stem cells or other progenitor cells, so that kind of on-target off-tumor toxicity could, with targeting some tumors or some targets, result in bone marrow aplasia, for example.
Yeah, that's, I guess, one of the challenges, since the B-cell CAR T and the myeloma CAR T are targeting antigens that are late in differentiation, so you're not killing the early progenitors, whereas AML, all the identified targets so far, I guess, are on early progenitor stem cells, so that's a huge risk. So tell us, your paper was very clever and interesting to read. So you decided to go with an approach that you call an AND gate. Why don't you tell us a little bit about that strategy?
Well, before I do that, I do want to say that while there are challenges, there are some major, I think, advantages or benefits to charge going after AML. Number one is it's a big unmet clinical need. There's been a lot of approvals recently for AML, but it's not really changing the natural history of the disease. Patients are still dying. And unfortunately, most patients diagnosed with AML will still end up dying of their disease. And also, AML is mostly a disease of the bone marrow. And CAR T cells naturally traffic to the bone marrow. So we don't have to worry about kind of the targeting widely metastatic solid tumor malignancies. AML lives in the marrow, and CAR T cells will always kind of be there. And while targets are a problem, it's been nice. We had 10 years of really good clinical development and evaluation of different targets for AML, as BiTEs or as antibodies or as ADCs. So we have some background already. So I just wanted to kind of say that it's not all doom and gloom with AML. There's some really good positives there. So what I wanted to do was, with this AND gate or kind of logic gating, is can I restrict killing to blast cells and avoid hematopoietic progenitor cells? So can I really kind of mitigate this on-target off-tumor toxicity with the design of this AND gate? And this is something that was developed and published by my mentor, Michel Sadelain at Sloan Kettering, where he demonstrated you could target two different antigens. In this case, it was like a solid tumor antigen and a hematologic antigen, something like PSMA CD19, if I recall correctly, and show that you can design CARs to work that way. But I wanted to take that from the artificial realm and adapt this to a real kind of cancer, and leukemia seemed like a great option for this.
Yeah, it does. Certainly an unmet need, and it's a great point. So that's often, I think, what researchers need to start with, right? Where is the potential biggest impact? So pursuing that a little bit further, in terms of the susceptibility of these cells to immunotherapy, to killing, since they're, I guess, more resistant to chemo, is there something intrinsic about them that gives them more resistance? And do you anticipate that they'll be susceptible to T cell mediated killing more so than other cells?
Yeah, that's a great question. And it's really hard to answer right now because we don't have much clinical success to be able to kind of differentiate why things work versus don't work. Right now, they just don't work. So really, we kind of have to extrapolate from the B cell field and the myeloma field with CAR T cells. And when we look at things like genomics, different disease factors, subsets, lines of therapy, overall, those things really don't correlate that strongly with outcomes. It's like the CAR T cell really just focuses on the target, and all those other features don't matter. So I'm guessing that that's going to play less of a role in terms of like things like AML subtypes and its chemo resistance. But I do think that it could impact T cell quality. So that's probably going to be the bigger impact. How easily is it able to isolate T cells from patients, expand them, and engineer them in a way where they are physiologically quite active? Because we've seen, again, going to the B cell space, we've seen differences between CD19 CAR T cells derived from patients with chronic lymphocytic leukemia versus those with B cell acute lymphoblastic leukemia. Disease factors can impact T cell quality. So that's probably one of my bigger concerns.
Right. So tell us about the targets that you picked.
So I focused on CD33 and 123. So when I started developing this project, I wanted to really focus on CAR design and as opposed to kind of target discovery, because that can take years. And what I liked about 33 and 123 is that their expression on blasts are well characterized, on hematopoietic progenitors and matured myeloid cells are quite well characterized, and we have clinical evidence of their therapeutic efficacy. CD33 is approved for ADCs for AML. There's no CD123 approved targeted therapy at this time, but there's been plenty of clinical experience with targeting that with antibodies and BiTEs, for example. So I felt they were well characterized, and I really wanted to start with those as my two kind of prototypical targets for AML.
Tell us about the levels of them on the AML cells relative to progenitor cells.
Well, it kind of depends on the studies that you're looking at, and if the cells that you are analyzing have been cryopreserved or if they're fresh. But in general, CD123 is probably higher expressed in earlier stage of progenitor cells, while CD33 is more kind of highly expressed at the later stage of myeloid cell development. And they're both highly expressed within blasts compared to their kind of wild type tissue expression. So those are some features that I felt made them amenable to this AND-gated targeting.
I guess the ideal scenario would be only one is expressed on any given normal cell, right? So that when both are expressed, that's when you trigger it. How close to that are these two? And are there any others that might be, pursuable in this fashion?
Yeah, I mean, there still is some overlapping expression of these within the progenitor cell population or the myeloid cell population, but they're not at the kind of the earliest stages of hematopoietic cell development. So we feel like the risk of bone marrow aplasia is less. But that's also why we developed the AND gate as well, to reduce whatever that risk is, to make it even lower. And there are, I think, other targets, CLO1, also known as CLEC12A, CD33, which is a target that I've also published on other papers. They have different tissue expression or bone marrow expression that can lend itself to potentially reducing some of that risk as well, even in OR-gated type of CAR designs. But those are things that are still kind of in development, and we don't have any clinical data outside of CD33 and CLEC12A. There's been some clinical experiences targeting that with OR gating, where they've seen quite good results, but many of the patients also develop bone marrow failure, requiring a stem cell transplantation rescue.
So for those who may not be familiar with OR and gating, can you just describe each of those for us and maybe tell us about the strategy used with the CD3 zeta and the 41BB?
Sure, it's essentially kind of Boolean logic, and or not. So if you design a bispecific CAR that's OR gated, that means if a cell expresses either of those targets, it's sufficient to be able to lead to CAR T cell activation. A not gated is that for a bispecific, again, is if you express either one of those targets, CAR T cell activation will be suppressed. And gating requires expression of both of those targets on the cell to mediate sufficient activation and target killing. So the way I've designed these CD33 and 123 CARs was to separate zeta from the co-stimulatory domain. And so this is also can be called a chimeric co-stimulatory receptor, so separating co-stimulation from zeta, which is activation. And so I have one CAR where I have zeta attached to the CAR, and I have the second CAR where I have only the co-stimulatory domain attached to the CAR. So that means when CAR A, for example, if CAR 1 is activated, that can lead to some activation of cytotoxicity, but not very good persistence, not very good expansion, not good cytokine production, because you're not getting that gene activation from the co-stimulatory domain. And if you activate only through CAR B or CAR 2, where there's the co-stimulatory domain, you get no activation at all. It's only when both of those targets are engaged where you get activation, cytotoxicity, co-stimulatory transduction, gene activation, gene regulation, then things such as remodeling, cytokine secretion, persistence, proliferation, memory phenotype differentiation. So that's kind of the idea behind this Boolean gate logic.
So I know a lot of this has to be empiric, and you tested tons of different combinations. Tell us about some of the different things you varied to test.
So well. First of all, started actually even testing different binders. So what I wanted to do initially was going, well, I know how we made our kind of first generation of cars against CD19, against many different targets. What we essentially did was look for hybridomas available in various kind of tissue repositories and use those to be able to sequence the binders and then kind of put them into a car. But each one of these antibodies were developed for a different reason. It could be to be a flow antibody. It could be a tissue antibody. It could be to be a therapeutic antibody. So just because they're an antibody doesn't necessarily mean it would be a good CAR. So my idea was, let's start from the very beginning, immunize mice against these targets, and screen a lot of different binders for their potential to be a CAR. So we ended up screening a few thousand different binders against for CD33 and 123 using high throughput flow cytometry. And we even kind of created this kind of functional CAR system where we put a JERCAT reporter, a JERCAT cell with a NF-kappa B reporter, and included an FC receptor in place of a binder for a CAR, and we co-incubated these things with our antibodies and looked, well, is this a good epitope? Is this sufficient to be able to activate our JERCAT CAR T cell? And so there's a lot of work that even went before we moved to the CAR design. So we ended up screening thousands of different binders before kind of moving down to a subset of a handful, something like 3 to 5 different binders for each target. So in addition to evaluating the different binders, which binders were attached to CD3 zeta versus the co-stimulatory domain, another thing that we looked at was how these combinations impact cytotoxicity and cytokine secretion. So wanted to look at beyond just one readout of does it kill this target or not, going, does it impact cytokine secretion? Are the cytokines being secreted classically associated with cytokine release syndrome? Are they super high levels, super low levels? Again, kind of thinking that if we decide to translate this, we want something that might have lower levels of toxicity. So those were the main different combinations that we looked at in functional assays that we used to evaluate these different constructs.
Yeah, so you did a whole lot of work in terms of in vitro testing, in vitro killing, cytokine production, as you said, and sort of came to a point where you were winnowing down all your different constructs to a very few. So what was your main criteria at the end there? Was it to select sort of a lead or one or two leads?
Well, we wanted to look at, does it kill? Yes. Does it secrete cytokines? Yes. But there's other kind of things, again, always kind of thinking of translation. Is this a product that we can put into a patient? What is this gene transfer rate? Do we get high levels of CAR expression within the T cell? But also, do we get high level expression of both of the CARs in the cell? Are they unimolar, 1-to-1 versus 1-to-3 or 3-to-1? Those are all kind of features that may not necessarily impact things like cytotoxicity or cytokine secretion, but could impact kind of the biological efficacy of the products. So we ended up really kind of settling on a few different constructs to kind of move into further kind of safety and efficacy testing in vivo.
I can't remember. Did you use two different like lentiviruses, each with the CAR on it, or did you have them in cis with like the protease domain to separate them?
Yeah, so we use a gamma retrovirus, not lentivirus, but as a quick screen, what we did was this kind of, I guess, mixed virus transduction. So we had CAR1 on a single construct and CAR2 on another construct that we used to make virus, and then we mixed those viruses together and use that to transduce T cells. That was more of kind of a screen because we had so many different combinations. We wanted to figure out which ones gave the results that we felt was promising that we then would create into a single genetic construct. And so we did both of those types of assays to kind of help us figure out which ones we wanted to take further for in vivo testing.
And does that single genetic construct give you pretty equal expression of both?
No, there's some variability. There's probably some genetic recombination that's happening for some of the combinations that where we don't see good to one to expression, but we did see on a couple of them good one-to-one expression.
Good. Okay. And then you went on and tested them in vivo. Tell us about those studies.
So I guess we wanted to look at two different things. We wanted to look at how well does it kill. So we didn't necessarily expect that this would improve on monoCAR, AML killing, because we had an end gate. So that means that there likely will be some blasts that express one of the targets, but not in both of the targets, and that wouldn't be sufficient. So what we were really looking for was is killing, similar? Is it kind of equivalent to monoCAR killing? And to do this, we looked at AML xenograft models where we planted NSG or immune deficient mice with these xenografts and looked to go, how well does it kill? And how well is that killing compared to a CD33 monoCAR versus a CD123 monoCAR? And we saw that the survival for the bispecific and gated CD33 123 CAR was slightly less than the 33 or 123 monoCAR, but it was not statistically different. So at least to us appeared that the killing was similar, at least significantly or statistically.
And then the second thing you were looking for was safety.
Exactly, safety. So we evaluated this in a couple of different models. One was we have these, standard colony forming unit assays where, based on work published by Sarah Gill, several years ago, we did a similar assay. We co-incubate the CAR T cells with hematopoietic progenitor cells for several hours, and then did these colony forming unit assays to count out how many cells were able to kind of go through the development process after CAR killing. And then we did an in vivo assay as well, where we did humanized CD34 stem cell engrafted NSG mice. So they had essentially reconstituted bone marrow and human peripheral cells, and then implanted or infused our CD33 123 gated CAR T cells, as well as the monoCARs into the mice. And what we did see was that the 123 monoCARs had the mice showed evidence of clinical toxicity and some cytopenias, which was reassuring because that's what's been published in the past, is that CD123 monoCARs can be associated with bone marrow toxicity. So we recapitulated that data, but we didn't see that with the 33 123 bispecific and gated CAR. So we felt that was, evidence that it had enhanced safety compared to a CD123 monoCAR.
Yeah, I think it's great proof of concept that your idea is working. I guess my question is it enough, right? What else needs to happen for this to translate to people? Because there was still some animals that the tumors seemed to break through, and so I guess that's my question, right? Is how are we there yet, or how much? I know this is a nice step in the right direction, but tell us about.
So that's a great question. So that's what I'm out when we start these projects. The end results of these projects are always, is this something that I feel comfortable with, getting an IND, opening for a phase 1 trial? Obviously, we would be doing that for safety, but part of this is we want to help patients. And I've taken a very kind of broad approach towards AML cell therapy. So I talked about this kind of early on in the discussion. Developed gamma delta T cells as a donor lymphocyte infusion for patients post-allo transplant at risk of relapse. We have the CD3 CAR that hopefully will be opening this year for patients with AML as well. And now we have this kind of N-gated CAR. And so looking for different areas where I kind of go, oh, this really is giving me some exciting results. This maybe is not so exciting. Can we potentially combine these in different ways? And so for me, I think this exercise was successful in kind of demonstrating that if you develop these N-gated CARs against biologically relevant tumor targets, you can make them work. You can reduce the risk of toxicity, but you're not improving necessarily leukemia eradication. And when you have a disease as heterogeneous as leukemia, that's probably not great, because it's going to leave residual disease that the patients will end up kind of relapsing from, and you end up with that problem later on. So what I'm looking at now is going, well, can I combine these 33 and 123 targets with 83 or with these gamma delta T cells to be able to improve leukemia eradication, and maybe move from the AND gate idea to more of the OR gate or other kind of different logic gating to improve the leukemia eradication and make sure there's less residual disease.
Yeah, I think it's a really important point that if it was easy, someone would have done it, right? And AML has been very challenging, and we need to marshal all of our different resources toward it. And it's nice that you're sort of having a holistic approach. What are all the different kinds of cell types can I target at this? One of the things that occurred to me while we were talking, I had not really heard of a NOT gate before, and I'm wondering, are there things that might be on the stem cells that aren't on tumor cells that you could use to sort of suppress the activity of the CARs against the normal cells?
That's a great question, and it's one that I don't think we've looked at, because the focus has really been on looking at targets. So looking for things expressed on blast and either not expressed on stem cells or expressed at very low levels. But that data is out there, right? There's been a couple of publications where people have gone through target discovery for AML to develop CARs, and likely that data might be able to be evaluated for looking for something highly expressed on stem cells or critical progenitor cells that we can create these NOT gates so that we won't have that toxicity. So that's a great idea. And so that's kind of the cool thing about this community of AML cell therapy researchers. We're kind of all working in our own space, but by publishing this stuff out, we kind of get to learn from each other and kind of figure out what are the most potentially beneficial trials that we can bring out there for our patients.
I guess my last question is, how long did this work take you so far? Because like I said, you made all kinds of different combinations, new binders, tested them in various ways. It looks like a ton of work, and it takes time, but just so our audience gets a sense of that.
Yeah, it took about 3 years in total. The antibody development was not as long as that I thought it was going to be. It was only kind of 6 months. But the sheer kind of work of doing all this screening, this high throughput screening, took about a year for us to be able to kind of whittle this down for those that then we kind of did good in vitro and in vivo kind of function studies. So it took a good 3 years. But the cool thing is that science and technology is progressing so rapidly. There's now machine learning and AI companies out there that can do some of the stuff that we did in 1 year or 1.5 years and get it down to something like 3 to 6 months. So I anticipate this type of work, that this approach that I took probably is not going to be the approach that I would take in the future. I would probably partner with these machine learning AI companies to reduce that amount of time to be able to really let us focus on the in vitro and vivo functional studies of those binders that we think are going to be most beneficial for translation.
Well, that gives us all hope for more rapid scientific progress in the near future, and your work gives us all hope for better treatments for AML in the future. So, and your paper was one of the more highly downloaded papers from our journal in a while. So we really appreciate your doing the work, giving us all hope for these patients, and sharing with us today.
Great. Well, I really enjoyed speaking with you, and I'm really kind of looking forward to how my other kind of collaborators and colleagues out there working in AML continue to move progress forward for our patients.
We look forward to that as well. Thank you so much.
All right. Take care.